Comet Christina, and welcome back for burps, belches, and bad breath. This is lesson 2, Mysterious Microbes. You're going to need your Mysterious Microbes kit, and then from home you're also going to need some water, some paper towels, some hand soap, because we're going to be doing some hand washing, some Scotch tape, something to mark with, so a pen, pencil, or even crayons can work for some of these, a permanent marker. A measuring cup is optional. You're also going to need your microwave, and then there's an activity where you can also use hand sanitizer if you have it. It's not necessary, but if you have it, there's an extra part that you can do as well. So go ahead and collect your materials, and then we'll get started. So today's lesson is called Mysterious Microbes. But what is a microbe? Microbes are very tiny living organisms that can only be seen with a microscope, and they are everywhere. Some microbes are beneficial, while others can be harmful. Most of them are pretty harmless though. So microbes do things like they help us digest and break down food. They help with things like breaking down dead and decaying matter. And some of them even help fight disease. Scientists that study microbes are called microbiologists, and they have discovered that microbes are everywhere, in the soil, in the air, even in our bodies. In fact, we have about 10 times more microbes in our bodies than we do cells that actually make up our bodies. Most of them do pretty good things and are necessary for us to stay healthy. So let's talk about some of the main kinds of microbes and show you some examples. Let's talk about 6 different varieties of microbes. The first one we're going to talk about is archaea. So archaea are actually microbes, and they're actually bacteria. But the difference between these and regular bacteria is that they don't have a nucleus. So they are the simplest form. And they're actually one of the earliest forms of life on Earth. Then there's regular bacteria. So normally when we hear bacteria, we think of bad things like illnesses. But there are really good bacteria out there too that help us do important things like digest our food. Next is fungi. When I think of the word fungi, I think of mushrooms because they are a type of fungi, and they can do anything from decomposing waste, breaking down dead organic matter, and they can even help with doing things like baking our bread. We use yeast when we bake bread, and it helps it rise, and yeast is actually made from a single-celled fungi. Next is protista. These are plant-like algae, and they actually produce a lot of the oxygen that we breathe, so they're super important. Then there's algae. These are really simple plants, and they can range in size from anywhere to so small that you need a microscope to see them, to really large seaweeds like the giant kelp. That can grow to be more than 100 feet long. The last one we're going to talk about is viruses. So on their own, viruses really aren't able to do much of anything. So what they do is they actually infect host cells in order to reproduce. And when that happens, it absolutely wreaks havoc and can cause disease. Coronavirus is the name given to a group of viruses, and this can include a lot of viruses, including even the common cold. So these viruses are packed in a capsule that has a spiked protein on the top of the surface, and it kind of looks like a crown when you look at it under a microscope. The word for crown in Latin is corona, so that's where the word comes from. So here is an image of, under a microscope, what the virus COVID-19 looks like. So let me ask you something. If you can't see something, does it really exist? The answer is yes. We just talked about microbes, the living organisms that can only be seen with a microscope, but they do exist and they're everywhere. And like we just talked about, some of those microbes are not good, and they can spread things like diseases and illnesses and viruses. So we do want to prevent the spread of those things as much as possible. In our next activity, we're going to be talking about fomites. Fomites are inanimate objects that can actually have microbes on them, and they can be used to kind of spread microbes from person to person or object to object. So we're going to be using a fomite and something called a medium to test out and be able to see the growth of that bacteria, because mediums have salts and vitamins and different things in them that promote the growth of those microbes so that we can see them with our eye. So let's go ahead and get started. For the Fun with Fomites activity, this is kind of going to be a two-step process, but I'm going to go ahead and tell you all the materials that you're going to need to get started on this one. There's going to be a little bit of a waiting period in between, so you're going to start this activity and then move on to activity 2, and then we're going to come back to it when it's ready. But you're going to need some water. From your kit, you're going to need the brown paper bag, the agar, your sterile cotton swabs, your spoon, the paper plate, the microwavable quart cup, and your 2 Petri dishes with the lids. Whoops. Also, you're going to need from home your Scotch tape. You're going to need 2 or maybe 3 paper towels. They should be pretty big because we're going to be folding them. So something like this size would be good. You're going to need a permanent marker or some masking tape, a pen or pencil, something to mark with. We're going to be using hand soap. This is the one where it's optional. You can use hand sanitizer, and then you can either use a measuring cup or you can use your 2-ounce cup. All right, so go ahead and get everything collected, and then we can get started and get this activity going. Okay, let's get started on our Fun with Fomites activity. So first you're going to take your quart cup, and we're going to add half a cup of water to this. I'm going to use a measuring cup. If you don't have a measuring cup, you can actually use your 2-ounce cup. You are going to fill this to the top twice. That would make 4 ounces of water, and 4 ounces is actually a half cup of water. So whichever is easier for you. I like to see the lines on the measuring cup, but if you don't have it, that's what that little 2-ounce cup is for. Okay, so half a cup of water into our quart cup. And now we're going to take all of our agar and pour it into the quart cup. All right, now we're going to take our mixing spoon and we're going to mix these together. You'll notice this kind of has a little bit of a funny smell. We're going to talk a little bit later about what this agar is made of, and you'll maybe understand what that smell is too when we talk about that. Okay, when it's mixed in really good, the next step we're going to be putting it in the microwave. The way you're going to do that is you're going to carefully take your paper plate, you're going to take one of your paper towels, you're going to fold it carefully on the plate, you're going to put your quart cup with your water and agar solution right in the middle of the plate, and then you're going to put the other paper towel folded in half on top. You're going to microwave this for about 1 minute and 30 seconds. It might get a little bit foamy, and that's okay. We do want it to steam. The cup is microwave safe, and I'm going to go ahead and get this started, and then I'll come back and show you what that looks like, and we'll move on to the next steps. Okay, so I just finished microwaving my agar solution. When you look inside of it, you should see no floating pieces of agar. Everything should be completely dissolved, and it is pretty hot. So this is where it's going to take some time between the beginning of this activity and the next one, because we need to pour our agar into both Petri dishes and let it cool completely before it can be used for the rest of our activity. So you're going to pour just a very thin layer into your Petri dish just so it covers the bottom, and then you're immediately going to want to put the lid on there because we don't want to expose it to a lot of bacteria, and there's bacteria even in the air around you. Okay, so I'm going to quickly pour this in the bottom. There we go. It's pretty covered. Put the lid on. Put that to the side. I'm going to do the same thing with my other Petri dish. Okay. So you just want it coating the bottom, just a little thin layer. And we're going to let that sit for at least 30 minutes until it cools down completely. So we're going to go ahead and move on to activity 2 and come back to Fun with Fomites. All right, so while our first part of activity 1 is getting ready to go, we're going to move on to activity 2. This is called Caught Dirty-Handed. You're going to need your lab sheet from your lesson. You're also going to need your black light from your kit and your Glow Germ lotion. From home, you're going to need some paper towels, a pen, a pencil, crayons, something to mark with, And then you're also going to need soap and some running water. So this one you're going to be using a sink to kind of wash your hands. So you might need adult help for this one as well. So go ahead and get your stuff collected and then we can get started. For this activity, we're going to be simulating with our Glow Germ lotion what it's like when you have germs on your hands, and then testing the effectiveness of different hand washing and hygienic practices. So it is important to note that you're going to be covering your hands 4 separate times. So while you do want it completely covered with the Glow Germ lotion each time between our different techniques, you do want to make sure there's enough for you to do it the 4 different times. So I'm going to demonstrate to you what it looks like. You want to do this on your non-dominant hand. So that means if you were to write on a piece of paper, you want to put it on the hand that you would not be writing with. So I'm right-handed, so I'm going to put it on my left hand. So you want to cover your hand completely. So you want to cover completely the front. You want to get in between your fingers really well. You want to kind of rub it around your nail area. And then we're even going to put it on the back of our hands. And then this is also why we need paper towels, because we're going to use this while we're washing our hands and trying our different techniques, but also because right now I need to get the Glow Germ lotion off of my other hand. If you need help with this, you can ask an adult to help you with this part as well. Okay, so now don't wipe off anything on your hand. You want it to stay completely covered. We're going to do a few different techniques to see what happens because when you use a black light on your hand in the dark, you can actually see the germs. So test it out as a control when it's completely covered. Go into a dark room. You can use a bathroom, just someplace that doesn't have as many windows as possible. Turn off the lights, and then for a control, turn on your black light and see what it looks like in the dark. You want to do this before we do any other techniques. And then we're going to go through a few different techniques we're going to try out, and we're going to use our lab sheet to track our results to see which is the most effective at getting rid of these germs on our hands. Once your hand is completely covered, this is going to be your control. Go into a dark room and use your black light, and when you shine the black light on your hand, it should be completely covered in our glowing germs. This is going to be how we compare how effective our different techniques are removing those germs. Make sure you use your lab sheet to draw a picture of what your hand looks like right now when you haven't done anything to try to wash your hands. Our first technique is we're going to run our hands under just warm water for 5 seconds. After those 5 seconds, you're going to blot your hand dry with a paper towel. Do not rub, and then go into a dark room and see if you see any difference. As you can see from my video, my hand looks pretty much exactly the same as when I hadn't washed at all. So make sure that you are tracking this on your lab sheet. Draw and see if you can see any differences in your hand from this versus when you didn't wash at all. For our next technique, we're going to run our hands under just warm water for 20 seconds this time. Again, use your paper towel, just blot it dry, don't rub, and then go into a dark room and use your black light and see your results. As you can see from mine maybe looks a little bit different than it did with the 5 seconds, but not a whole lot different. And then once you're done and you track your results, you're going to cover your hand again in the Glow Germ, and we'll start over with the next technique. Okay, so this time we're going to use soap and warm water, but for only 5 seconds. And again, just blot your hands dry. As you can see from my video, mine maybe looks a little bit different on the front of my hand, but not so much the back, and not really at all under my fingernails. So go ahead and track your results too, and then when you're done, you're going to cover your hand again before our last technique. So for our last technique, we're going to use soap and warm water for 20 seconds. You're going to scrub really good and then again blot it dry. So you can see here my hand looks pretty clean from those 20 seconds. So I think it's safe to assume that 20 seconds with soap and warm water is a really effective way to remove some of those germs from your hands. So use your lab sheet and track your results and see how yours compare. Did 20 seconds in warm water work best for you as well? All right, now my agar is completely set, so we're ready to move on and finish our first activity. So the first one we're going to do is you're going to take a little bit of water and add it to your 2-ounce cup. All right, next you're going to pick a household object or a foamite. So anything like a cutting board, you could use your laptop, the keyboard, a cell phone. You're going to take one of your sterile cotton swabs. You're going to peel it open very carefully because you don't want to touch the cotton end of the swab on anything. So you're going to pull that out carefully. And you're going to dampen the end of this in some water. Then after you've picked your foamite, you're going to take this and you're going to swab it against the surface of whatever you picked. I'm going to pick my cell phone. So when you go along the surface, you want to kind of turn it as you're rubbing against it because you want to kind of get some of the surface covered on every side of your cotton swab. Don't touch the end of your cotton swab, only touch it to your object. I'm going to put it around all the different parts, not just one part, so maybe around the buttons a little bit where I touch. If you're doing like a keyboard, you want to do the same thing. You want to move it around different parts of the object. And then you're going to open your first Petri dish. And this is important, guys. You don't want to use too heavy of pressure. This is going to be kind of gentle. You don't want to break the surface. And you're going to do a squiggly line all across the Petri dish. So you're going to want to get this swab kind of all over it. But again, be very careful. Don't press down too hard. And you're going to want to move this around to your cotton swab to kind of get all the different sides of the cotton swab touching your Petri dish. This is the one that if you have hand sanitizer, you can put a little hand sanitizer over one of the parts that you touched and then make a hypothesis. Do you think that the growth of the microbes is going to be different on the place that has hand sanitizer versus the places that don't? Once you're done, you're going to cover the lid. You're going to take 2 pieces of Scotch tape and you're going to tape that lid shut, because we don't want it to be exposed to anything while we are setting this aside and kind of watching the magic growth happen with our microbes. And you also, when you store this, you want to make sure it's stored upside down because there's going to be some condensation and we don't want that to run onto our agar. We want that to go down into the lid. Okay, so I'm going to set this to the side and now we're going to do our second Petri dish. Before you open it, take your magic marker and I'm going to draw like a cross shape right across the middle. I'll show you what it looks like. Just like that, and then we're going to label these. So one side is going to be a T for thumb, one is going to be a W for water, one is an S for soap, and one is a C for control. So it's going to look just like this. And I'll explain to you what these markings mean in just a second. So we want to do that before we ever open it. So now what you're going to do is you're going to be using your thumb for this one. And you're going to just make an imprint in the agar on these different sections of the surface, your medium. So the one here that says thumb, you want to do this with a thumb that you've used after you touch a bunch of surfaces, because we did just wash our hands for the glow germ activity. So you want to touch other things, try to touch different kind of foamites and get those microbes on your thumb. Then you're going to press your thumb on that section of your agar. Then you're going to wash your thumb with just water, pat it dry, and stick it in the W section. That's going to be what it's like with just water. Then for the S section, that means soap. So you're going to wash that thumb with warm water and soap, just like we did for the glow germ activity, and press it in that section of the Petri dish. The C just means control. So control is like our stable testing. So we're not going to touch it at all with our thumb. We're going to compare our different thumbprints to not touching anything and see what that looks like. There is microbes and stuff in the air, so you might see some growth. It's possible. So we're going to see what that looks like. So when you're done with those, you're going to take your Scotch tape again. You're going to tape those on the side. Make sure with your lid you're not twisting it so that you can keep track of what each of your corners are. So where you touch, you want to make sure that that's labeled correctly. So you're going to tape it on both sides. And then again, this one you're also going to store it upside down for any condensation, okay? And then you're going to take both of these Petri dishes and you're going to put them inside of your brown paper bag. So set them down on the bottom. And when you want to place them somewhere kind of warm, not super warm, but kind of warm. So in an area that maybe there's like a heater somewhere nearby, that would be a good spot. The top of my refrigerator is a little bit warm, so I'm gonna put mine up there. And then you're gonna give it at least 3 days to kind of sit there, let the magic happen before you open it up and kind of see that growth. And then after those 3 days, you can kind of check it out on a daily basis. Put it back in the brown bag, seal it up again, and then check it out every day and see if you have any new growth. It's really important though that you give it that 3 days to kind of sit and let it do its magic. So when I have my finished results, I can show you. But for now, I'm going to show you just an example of a Petri dish that has some bacteria growing on it and see how cool it looks, because yours are going to look pretty similar. Now we're going to do activity 3, eyelash creatures. From your kit, you're going to need your other plate, your other paper plate, your colored beads, your 2 googly eyes, and your modeling clay. And then from home, you're going to need anything you want to kind of color with. I'm just going to use a pen. You can use crayons, markers, anything like that. So go ahead and collect your materials and we can get started. We're going to make our own version of a demodecid. So what's a demodecid? They're super tiny mites that actually live inside the hair on your body. So anywhere from your head to your face to inside your nose to on your eyelashes. They're so small they can only be seen with a microscope. They're about 0.1 to 0.4 millimeters long. And a female can lay up to 25 eggs on one hair follicle. So that might creep you out, but everyone has these mites living on their body right now, and they're completely harmless. So the first thing you're going to do is you're going to take your plate and you're going to draw an eye. So you can be as creative and colorful with this as you like. I'm going to keep mine pretty basic because I'm not really the best artist. So I could spend a lot of time putting a lot of effort in and not really get that far. So have as much fun with it as you like. Make the eye whatever color you want. Maybe it's your eye color, maybe it's an eye color you think is really cool, an eye color you'd like to have. Just make sure you draw an eye and that you include some eyelashes because that's where our demodecid is going to go. So here's my eye, nothing fancy. And next we're going to take our modeling clay and we're going to make a clay model of a demodecid. So it should be like a long kind of shape, and then they are kind of smaller on one end than the other. So let's shape this out. The tail is thinner than the body. So you want to make the head a little bit wider than the tail. Mine's going to be a pretty big one. I don't know about yours. Okay, so there's my demodecid. And before I put it anywhere, I need to give it little legs. So we're going to actually use our beads, our colorful beads. So you're going to have 4 on each side. I'm actually going to stick mine in sideways. I want them to stick out from the body rather than short ways, but you can do yours however you like. It says in the materials that you might need glue. I have found that I did not need glue to make any of this stuff stick. I could just stick it right into the clay and it worked just fine. So I would say glue is optional. Okay, so there we go. I've got my 4 legs, and now I want to add some eyes to my demodecid. So I'm going to use my googly eyes and stick them right on there. And then I'm going to put that right into my eyelashes. where this thing lives. So there you go. There's my Dimodicid. It's pretty colorful, right? It's got like 8 different color legs. And it's just living happily in my eyelashes. I wonder if it's a female one and it's gonna lay eggs. Ew, right? Okay guys, well I hope you had so much fun and that you learned all about microbes. Now you know all the different kinds. We covered a lot of different kinds of microbes, and we learned a lot about spreading of germs and how to prevent that, and good hygienic habits for hand washing and things like that. So I hope you have fun, and I'll see you at lesson 3 of burps, belches, and bad breath. See you guys.